Hello, and good morning, good afternoon, good evening, everybody. I am Sohela from Ingenuity Systems. We're part of QIAGEN Bioinformatics. And today I will be talking to you about using Ingenuity's Variant Analysis, a platform that can be used for many different types of analyses for your sequence variant data. So to start out, I'm going to give you a quick introduction of the platform and the underlying knowledge base that we're going to use to analyze the different data sets. And then I'll present 4 different case studies where we have used the application to narrow down to the candidate variants for as either causal mutations or as targets for therapeutics. So, variant analysis, in a nutshell, is a secure web-based platform that is used for interpreting human genome data. It is a flexible platform for annotating and comparing different genomes. It allows for scientific iterations, meaning that if you decided to change your mind about specific criteria or comparison steps that you're using, you can rerun those in an iterative fashion within minutes. It leverages the deepest functional knowledge base that is available, which is the Ingenuity knowledge base, and it also includes very rich cancer-specific annotations for analyzing somatic data. It has robust analytical and statistical methods that are built in for different types of studies, so either you're just comparing a bunch of probands to controls, or you could have tumor normal samples, you could have large kindreds or big cohorts with quantitative data and so on. And lastly, it's a platform where you can share your samples or analyses. So if you have a collaborative group or you would like your colleagues to look at what you've done using the application, you can just share the analysis with them. So where does it all start? And it starts with the knowledge base. And Ingenuity's knowledge base is this database of information that is collected from a peer-reviewed literature as well as third-party databases. And all the peer review is done manually. So we have a PhD-level scientist who reads the paper and then adds the finding into the knowledge base. And then this knowledge base becomes the foundation for our applications where you can analyze data using variant analysis or our more popular and widely used application, IPA. Specifically for variant analysis that was launched in 2012, we have curated, aside from the manual literature findings, about 350,000 manually curated human phenotype mutation associations, as well as over 20,000 curated disease models and almost 200,000 pharmacogenetic findings where we have information about genes that are targeted by specific drugs, as well as variants that are used to determine those targets. So aside from our manual curation, we also have information from third-party resources such as Cosmic, TCGA, OMIM, MGI, and others. And we have in silico analysis tools such as SIFT, PolyFan, and Vista tools that are built into the application to help with the analysis. So in addition, QIAGEN also acquired BioBase, which is all part of QIAGEN's bioinformatic unit, and BioBase brings HGMD, which is the most commonly used human genetic mutation database, and we have now access to this database through variant analysis. So with a subscription, you have full access, and without a subscription to HGMD, there is still access to the free version through the application. So what can you do with this knowledge and the application together? Well, you can use this to analyze data sets with gene sequence variants for a number of different types of studies, whether those are studies looking at rare Mendelian diseases in trios or quads, or if you have large categories that you're looking at. You could also have a small cohort data set, so there's a small number of cases that you're comparing to a small number of controls, or you could have large data sets where there are a large number of cases and controls, and what we're introducing shortly is the ability to do quantitative trait analysis, where you will be able to look at a variable trait over a bunch of samples. In addition, we are able to do tumor normal comparisons to focus on somatic variants by removing variants that are also present in the germline. So now I'm going to present an example of each of these 4 case studies that you can see, and you'll be able to see how we're able to analyze the data and get to our causal variants or pharmacogenetic targets. So starting out, I'm going to present a trio where we have a family where we had a proband, a 5-year-old who presented with medulloblastoma. He had a 27-year-old normal parents with no disease or history of medulloblastoma. In addition, he had a sister who was diagnosed with acute myeloid leukemia and passed away at the age of 3. So we sequenced the proband and his parents to determine what it is that was passed from the parents or acquired de novo in the child that led to medulloblastoma. With that, I'm going to now present the analysis of the variants uploaded into the platform, and I'll go through the different filters and analysis settings that were applied. And just for the future studies, I just want to point out that I'm only going to talk about the confidence, common variance, and predicted deleterious filter in this particular example, and then mainly just skip to the genetic analysis and other filters for the subsequent studies. So starting out, we looked at this data set. We uploaded the variants from the proband and the parents into the application and had about 6 million variants in the 3 samples. We then used the confidence filter with the aim of removing any variants that do not meet the quality cutoffs. So these are mostly industry standard cutoffs, such as quality of 20 or higher, read depth of at least 10 or more reads lining up at the variant position. And also we have Ingenuity's built-in quality features where we're able to remove false positive variants. So these are variants that if you have sequenced any exomes or whole genomes, you've probably seen these in genes like mucins or HLA or Titin. These are mainly false positives that are present due to the large gene families or large genes themselves. So we can remove them by looking at exonic windows of 100 base pairs and determining the top 5% most highly variable regions from the 1,000 genomes, as well as determining the top 1% of the most significantly mutated genes or variable genes from the 1,000 genomes. Now, all of these settings in variant analysis where you see a little box with arrows are changeable. So if you wanted to have a higher quality filter, you could set that to be higher. If you wanted to set your variable frequencies to be lower, you could set them to be lower, or if you wanted to be more stringent, because your platform has much more false positive data, you can actually dial these up and down. Next up, we applied a common variance filter. So anybody who's doing a sequence variant data analysis is aware that for the most part, we're not interested in common variants, and we want to focus on variants that are either never seen before or only seen in a very small percentage of the normal population. So for that, Ingenuity has built-in ability to compare your variant data to the 1,000 genomes, complete genomics publicly available data, and the NHLBI data set, where you can set a frequency cutoff. So here I'm only focusing on really rare variants by looking at less than 0.1% variant percentage in any of these databases, again, depending on the phenotype, the commonness of the occurrence of the phenotype, we can dial this frequency up and down. And as I mentioned before, this can be done in an iterative fashion. So if you set the frequency to 2%, change your mind, come back and want to dial it down, you can do it and apply it right away, and it's executed within minutes. The next thing that we would be interested in is determining which of these variants actually have a consequence on the gene expression or protein function. And again, we can leverage Ingenuity's knowledge base for that information, or we can also use a number of in silico tools that are available to determine whether a variant would be damaging to the gene or protein function. And we can do these separately, or we can apply them all together as an OR filter. So up top here, in the first section, we're looking to see which variants are likely to be present in previously known databases such as OMIM or TCGA or COSMIC, or have been established in published literature to be causal or damaging. And again, we apply ACMG guidelines to determine whether these variants would be pathogenic, likely pathogenic, uncertain significance, or benign. Similarly, we can use literature findings as well as in silico tools to determine whether a variant may result in gain of function. So, for example, certain variants in promoter region may result in increased expression of the gene, and so we can see if that variant is previously defined from the knowledge base, or we can use our analytics in the Ingenuity analytics or BSIFT algorithm to determine their gain of function. Furthermore, if none of these criteria are met, if we want to determine whether a novel variant is possibly causing disease, we can look to see if it's a frame shift, in-frame deletion, or it could be introducing a start or stop codon, or we could look to see if it's a missense variant and further check to see if it's damaging based on criteria determined by SIFT and PolyFem. We could also look for splice sites, and in addition, we can look to see if there are any regulatory changes in the mutation due to these variants. So look for microRNAs, structural variants, promoter losses, enhancers, and also UTR regions. So after we have applied these three filters, you can see that we're already down from almost 6 million variants to about a little over 1,000, so less than 1,500 variants are left. At this point, we would now apply our genetic model for further removing variants that may not be causal. To do so, we have a filter called genetic analysis, and what genetic analysis is able to do is apply your specific genetic model. In this case, I had uploaded a pedigree to designate the relationships of the parent to the child, and so what I can now do is only focus on variants that meet the Mendelian inheritance criteria. And because this is a child who is born to two normal parents and has the disease, and because his sister also has the disease, I'm sort of ruling out the hypothesis that this could be de novo and focusing on inherited variants with a recessive model. So I'm only looking to see if there are any causal variants that could be either homozygous or compound hets, and removing any variant that's a compound het from, a homozygote in the parent. So once we have set our genetic analysis filter and we have reduced the number of variants that meet that criteria to 16 in about 9 genes, I then can go on and apply the biological context to this analysis, wherein I am setting the criteria for the disease. So I'm looking to focus on variants or genes that are previously associated with medulloblastoma and acute myeloid leukemia, either or both. And also looking for genes that may be one hop upstream of the known genes, meaning in the knowledge base, we have information about genes that are directly associated with medulloblastoma and acute myeloid leukemia, But we also know genes that may interact with those known genes, so we can go look 1 or 2 hops upstream and also 1 or 2 hops downstream. So starting with a more conservative approach, I'm looking only 1 hop upstream. At this point, I'm left with 5 genes with 10 variants, all of which meet a compound heterozygote criteria. So if you look at this list, I have several genes that are, several variants that are in this child and inherited from at least one of the parents. I automatically rule out the first one because both the variants are coming from the same parents. It's likely not a real compound het, and start with the second gene, which is GREM6, which looks like a compound het with 1 allele inherited from each of the 2 parents, and start to investigate it further. When I click on the row for GREM6, I get another window pop up on my screen that lets me have more information about the specific variant that I highlighted, but has an option to look for paths to phenotype. When I click on paths to phenotype, I'm able to see how GREM may be related to 1 or more of the biological context criteria that I specified. So here it looks like GREM is 1 hop away from acute myeloid leukemia, which was the trait that was found, or the disease that was found in the sister. I can start to read more about the relationship by clicking on these vertical lines that connect the 2 genes and also connect the gene to the disease. And by clicking on the first line between GREM and its target, I can pull up the references that make that connection. I can click on these links, go to the abstract in PubMed and read the publication. Similarly, I can click on the second line below and read publications that connect the target to acute myeloid leukemia. However, while this looks like a good target, I wanted to further explore other targets because one of the 2 variants for GREM6 is called out to be tolerated or benign by SIFT and polyphen. So I move on to my second candidate in the list, which is PSM2. Both of these look like good possible causal variants because one has a splice site loss and the other has a stop gain. So when I start to investigate PSM2, I click on the paths to phenotype for this gene, and what I find is that there are lots of relationships between a number of genes associated with acute myeloid leukemia as well as medulloblastoma and PSM2. Again, I can further investigate these relationships to determine how believable and robust those are. And finally, based on more digging, we were able to establish that PSM2 is the candidate that caused the disease in this particular child, and possibly also his sister. So this was one example where we looked at a trio to determine inherited disease variants, and by taking into account family relationships between the parent and the child, looking for Mendelian inheritance, and then applying the biological context to look for the appropriate genes. I'm going to now present another example where we looked at a small cohort. In this example, we're looking at a disease called pheochromocytoma with 3 probands from extended pedigrees with a family history of the disease. So each of these cases presented with pheochromocytoma and had family history. And we took these 3 cases, sequenced our exomes, and compared them to 7 HapMap control samples. So these 3 males and 4 female samples. These findings were initially made in 2011 and published in Nature Genetics. However, we were able to get the sequence data and independently analyze it using variant analysis and reach to the same conclusions as the publication, which did not use variant analysis, just to be clear. So we get the data, we upload it, and from these 7 control and 3 case samples, we had almost 26 million variants that were uploaded. And we applied our confidence criteria for looking at good quality variants. We applied the common variance criteria to only focus on variants that are not seen in the normal population. So we actually dialed the frequency down to 1% in this example. We applied our predicted deleterious criteria to look for known mutations as well as look for variants that would cause damage to protein function. And then we went ahead and applied the genetic analysis criteria. So in this case, first off, I wanted to select any type of variant, whether it would be homozygous, heterozygous, compound het, or any other criteria in the cases, and remove any variant that was present in the control. So we remove every variant that would be in the control. In addition, I wanted to make sure that since these three cases have the same disease and a likely inherited disease, I wanted to see if it's possible that all three of my cases would have a variant in the same gene. So we set the filter up to look for any variant in the cases that are present in the same gene for all three of them. So at this point, when we apply this filter, we're left with 24 genes with 35 variants that meet our criteria. I then go ahead and apply the biological context, which in this case is pheochromocytoma. So setting the biological context in variant analysis is very simple. You just go into the biological context filter and type in the name of the disease or phenotype or pathway or gene, whatever it is that you would like to focus on. And then in this particular example, you'll see that we'll find a direct link between our candidate and the disease. But when we first discovered it, we had actually used 2 hops from the phenotype to look for possible novel candidate genes for the disease, and also look to see if there were any genes that were 1 hop below the known genes that are involved in pheochromocytoma in the pathway. So when I apply this criteria, I get 19 genes with 26 variants. Now, as you can see on my screen, several of these variants seem to be present in all 3 samples. So it's the same variant that's in all of the 3 cases. When we further investigated the population frequency of these variants, we found that each of these variants were rare, as in never seen before. So going with the likelihood that a novel variant would be present in 3 cases all at the same time being somewhat rare, we decided to first be a little stringent with our criteria and apply another genetic analysis filter. So you can stack genetic analysis filters or any other filters that you're using to build the pipeline for analysis. And what we decided to do here is we said exclude any variant that's present in all 3 of the 3 cases at the variant level, meaning we'll remove any of the variants that look like this. When we go ahead and do that, we get a slightly cleaner table where we have 7 genes with 14 variants in all our cases. At this point, we have a small number of candidates on our screen, and we can start investigating these by looking at paths to phenotype. The only one that I'm going to show you is the one that was the published candidate, which was also then validated using segregation analysis in the families. So when I look at the candidate Max, which seemed like the most plausible candidate at the time, we click on it and we find that Max is known to be directly associated with pheochromocytoma. So you can find direct links between genes and diseases. And then we can click on the connecting line between the gene and the disease to investigate further by looking at entries in PubMed, and so we found that there were a number of entries that connected the gene. We also found ClinVar and OMIM entries for these particular variants. That's because the gene has now been characterized and published, and we curated the paper that published these findings initially. But to give you a flavor for how the knowledge base actually found it, I want to do a little flashback. and show you how we were able to find this variant independently of the paper and the curation of the finding by leveraging the knowledge base. So when the paper was not published and we hadn't curated the relationship between MAX and pheochromocytoma, we were able to look up any genes that are associated with neuroendocrine carcinoma, because pheochromocytoma is a type of neuroendocrine carcinoma. Look at the genes that are known to be associated with pheochromocytoma from the knowledge base, and then we found MAX as an interacting partner of those genes, and were able to establish that MAX was a possible candidate for the disease. So that's another way of looking at different types of data sets where we're doing a case control comparison, removing variants that are in the cases. This can be done when you have a small number of cases. But there are scenarios where you would have a large cohort, so you have a large number of cases and a large number of controls. We're talking hundreds and thousands of cases and controls. And for these types of studies, we will leverage Ingenuity Variant Analysis built-in statistical association tools. And to do so, I'm about to present to you a study that was done using samples from the Personal Genome Project. The Personal Genome Project is a project where volunteers enroll to sequence their genomes. The data is available through their website, thepersonalgenome.org, along with phenotype data for all of these subjects. So we went through the data set and found 66 cases with myopia and found 45 controls to match to these cases and try to identify genes and pathways that may be enriched by variants in our myopia cases. So moving on, we took the data, uploaded it, and again, you can see here we have a large number of variants uploaded to the system. It's over 2.7 billion variants from these nearly 100 cases that we have in the data in our system here. We use the confidence criteria to remove all variants that do not meet the quality cutoff. And then here we did something different, and I wanted to do this differently to show you that filters in variant analysis do not have to be in a specific order. You can decide to change the order of these filters as you wish. So we first looked to see which variants would be functionally relevant, and again here, instead of using the default name predicted deleterious, I changed the name to functionally relevant, just so I know what I was doing in that particular analysis. We applied the criteria to look for variants that may be previously known to be causing disease, as well as variants that would cause loss or gain of function of the gene. I then went on to remove the common variants by applying the common variance filter, and here I called it variants because I'm only focusing on variants that are in 5% or less of the population. And then I went to apply, instead of applying the genetic analysis filter, I used statistical association. And what statistical association filter has is it has built-in tools to apply different statistical models for your analysis. So one you can do is a burden test, which is up top here. And with the burden test, we can look for burden of variants in either genes or pathways. So here I'm doing a gene burden test. And we can look for burden of variants in either cases, controls, or both cases and controls using the SCAT-O algorithm that's built into the application. In addition, if you actually just wanted to do a common variance association test, we also have the test built in here using Bonferroni or Benjamini-Hodgberg corrections for multiple testing, and this will just see if there are specific variants associated to your cases or controls, just like you would do in a GWAS study. So once we apply, in this example, we are looking to see what rare variants may be enriched in specific genes or pathways, in this case genes. I go on to the next step, and then I actually get 11 genes with 73 variants that are enriched in the cases. And then I went ahead and applied all biological context relevant for myopia, eye disease, eye development, eye morphology, just to be as broad in my analysis as possible. And I got 5 genes with 29 variants. So you can see that on my screen here. I'm looking at the genes table with the 5 genes that are linked to myopia as a result of my burden test. And here's the list of variants and number of cases and controls that carry those variants. So now I can start examining each of these in more detail to see how they may be associated to myopia. And here's the list of the genes with the specific variants. I'm going to show you the first one, which is the IFAC gene. In that we were able to see that it is associated to a number of phenotype and myopia-related disease conditions through a large number of known genes that cause these disease or development of eye. What I was more intrigued by in this analysis was the HK3 gene, because as you can see here, HK3 gene seems to have an enrichment of this variant in most of its cases. Initially, I was tempted to dismiss this as a sequence artifact, but then when I found that there is actually a population frequency of 1 to 2%, it seemed like a more of a real enrichment. So here we have a missense variant that is enriched in most of my myopia cases. And when I investigate the gene, it looks like a good candidate as it's involved in morphology of eye and eye development through these 2 known genes that are associated with the phenotypes and pathways. So that made it a good candidate, and here you can see a view of all the cases with the variant, the quality, and we can see that these are all good quality calls, so this is likely not a sequence artifact that may have been observed. So that was a study where we were able to identify a candidate for a common phenotype by using a statistical sequence in a larger number of cases and controls, and a statistical burden test to look for enrichment of variants in cases. Before I move on to case number 4, I'm just going to quickly see if there's a question that I can address. So there is a question that's asking about normalizing for DE-seq and EdgeR algorithms as input. That is not currently built into the application, but if you had values associated for those as vet files for specific variants, you can upload them into the application and filter that. So last case that I'm going to present today is a paired tumor normal study, and for this we're using 2 cell lines for breast cancer, and we have 2 breast cancer cell lines matched with their normal cell lines. So you can see these are the tumor cell lines, and these are the normal cell lines. And for somatic variant analysis, what we do is we match samples by their subject ID. So that would suggest that these two samples are from the same individual, and then those 2 samples are from the same individual. And this way, the application pairs them and only removes a germline variant if it's present in the matched normal and not in the other normal. So once again, we sequenced these with complete genomics. So we got whole genome data, and we were able to then go ahead and filter for confidence, common variants, and apply the genetic analysis filter, which I'm going to show you here. So here we applied a somatic variance filter. So this is a tumor-specific filter. And what this is designed to do is, first off, pair match your samples by individual. So each of those 2 tumor normal pairs got matched to each other. And we then went ahead and removed any variant that was present in the matched normal from the analysis. And this was done so that only if the variant is present in the matched normal and the tumor will it be removed, otherwise it will be retained if it was a somatic variant only. We then had the ability to apply a cancer driver variance filter. So this is a special filter that I was talking about earlier which is designed specifically for analyzing cancer data. And what we have here is the ability to look for variants in genes that are associated with cancer knockout models in mice that are known to be associated with processes and pathways for cancer, as well as if any genes are specifically known targets for therapeutics, any published findings. Also, we are able to include information from COSMIC and TCGA to this variant analysis. Once we apply the cancer driver filter, we're now focusing only on the 1,500 plus genes that are known cancer drivers or those that interact with these known cancer drivers. We then apply our specific biological context for breast cancer and are able to reduce the number of genes to 209. That's still a lot of genes, especially if I was interested in focusing on therapeutic targets. So I can do further analysis to narrow down from this list of 209 to those that would be relevant for my study. So first off, because in my predicted deleterious filter, I had applied the criteria to include anything that was likely pathogenic and uncertain significance, I now want to remove anything that is non-coding. So first off, I'm removing anything that's in a promoter or in the UTR, and I'm doing this by applying a second predicted deleterious filter where I set it to exclude promoters and UTR variants. So that will reduce my list of genes to 87. So these 87 genes all have coding variants. Next up, I want to focus on those that are targets for pharmacogenetic findings. So if there is a drug that is known to target the specific gene or variant, and I come down to 44 genes, and this is done using the pharmacogenetic filter, where I'm looking for any genes that are associated with response, toxicity, or metabolism, or targets, or any other drug binding relationships that are known. I can also type in name of specific drugs to further limit this list to those that are known targets of that drug. And by doing so, I now go down to 44 genes, and I can then investigate each of these variants in genes by clicking on the pharmacogenetic finding to see which drugs were used, how those drugs were associated to that particular gene or variant, and then these can be used as therapeutic targets, either for research purposes or in the clinical settings to see which are the best drugs to be applied. So with that, I would like to end the talk and just do a little quick recap. So what we saw here today was an application that was used to capture either causal variants or targets or variants or genes associated with different diseases or phenotypes. And to recap the key features of the application, it's a very secure application, so all data that you upload is stored on our secure servers. It is not residing on your laptop, it's residing in our servers that are both HIPAA compliant and have safe harbor. You also have the ability to do a really fast and interactive filtering and analysis of your data by applying quality cutoffs, common variance cutoffs, focusing on causal variants that would have a deleterious effect on the gene, and then really put those variants into the biological context. We can also then share our analyses with our colleagues, so you can either share uploaded samples with your colleagues, or you can analyze the data and then share the analysis with your colleagues who can review it, make changes, and initiate collaborative discussions for the data that you look at. And lastly, it's a very supported product, so if you have any questions or concerns about your analysis, understanding what you look at, or any technical issues, you can reach out to Ingenuity for technical as well as scientific support. And with that, I'm going to end and take any questions that you all may have. No other questions? Okay, so since we have no questions, I am going to end the session. If you do think of something, please feel free to reach out to us at support@ingenuity.com. Oh, I see a question popping up. Oh, great questions. The first question I have here is, what are the input formats for variant analysis? And so variant analysis will take your VCF files that are generated through the GATK pipeline. It will take complete genomics, master var, and high confidence junction files, as well as Illumina GVF files, the genome variant files. And how long does it take to complete the analysis? So the length of the course of time taken to complete the analysis depends on the number of samples and the complexity of the data. So with a smaller data set, about 100 or a couple hundred, usually you're done within 15-20 minutes to an hour. Larger data sets of thousands of samples, especially if there are thousands of whole genome samples, it may take a while, a few hours for them to be done. Either way, once you set up your analysis, you can log out of the application, and you will be notified by an email once your analysis is done. So I have a question about the HCMD database. Could you be more specific about what you're looking for? So the HCMD database basically has information about known genetic mutations that are associated with disease phenotypes. So what we're able to incorporate in Ingenuity Variant Analysis at this time is, if you have a variant in your data set that's also present in HCMD, you will see a link to that variant. So let me go back to one of my slides and show you where you will find that information. So this window, when it pops up, if you have a variant that's also present in HCMD, it will be indicated with the HCMD accession number just down here. And then you will be able to click on that link to read more about that variant from the database. If that variant is in the free data set and you don't have a license, however, if you have full access, you will be able to get all the information there is in the HCMD database about all variants. The future plan for HCMD is, with regards to variant analysis, is to incorporate it in the application, so you will be able to filter your variants against what is already present in the database. There's a question about applying these features to analysis of other organisms. Unfortunately, at this time, the application is only able to take and analyze human genome data, so we're not able to analyze other organisms. Accuracy of the analysis, that's a good question, but so accuracy is really, it's a bit of a subjective question. What we're able to do with the application is really get you as much information about the variant to make a call whether that is a good candidate or not. So we're able to put it into the context of the disease and the pathway, and also we're able to apply analytical criteria such as if it fits the inheritance model that you would expect it to fit. And as far as the analysis is concerned, it picks up the variants accurately. So if a variant was transmitted from the parent, it will be appropriately filtered into your data, and you will be able to analyze it further. As far as the biological context is concerned, the knowledge base is as accurate as the actual published literature is. So if there is published information about a variant, there will be more information. There will be accurate information about that variant in the database. I hope that answered your question. So we do run internal algorithms as well. So sorry, so the next question that I have here is all the information from the knowledge base, or do we run internal algorithms? So we run internal algorithms to determine what variants should be retained for further biological analysis. So for example, our algorithms will determine whether variants are causing gain of function, or we're running algorithms to determine if the variants fit the inheritance pattern that you're looking at. So if you were trying to see if a variant was coming from the parent, or whether it's a somatic variant versus a variant that was inherited from the germline, or if you have a large cohort and you're trying to see all the cases have a burden for a variance in a particular gene, all those analyses are derived through internal algorithms that Ingenuity has built in. So there's a question about a free trial version. We have a 30-minute free preview for your data. So if you upload your data into the system, you can set up an analysis, and once the analysis is done, you will get 30 minutes to go through all the filter settings and get to your list of candidate variants. This is to give you a flavor for how the application works. If there was more interest in trying it out, we do have nominal charges for paid pilots to test out the application. And if you're interested, you could email us and we can set you up with the right person to talk to. Okay. More questions? Okay, so if there are no more questions, we can end the session. And thank you all for your time. And please feel free to reach out to us if anything else comes up after this. Thanks.